All right, this is going to be our lecture on electrolytes, and it's a pretty short lecture, so let's go ahead and get on with this. Hopefully you have your notes available because part of this involves reading. First thing, body fluid, water and dissolved substances that comprises 45 to 75% of the body by weight. Now I've given you a breakdown here. Intracellular fluid, this is like inside of cells, the cytoplasm inside of cells, about 2/3 of your fluid volume. Extracellular fluid, which is outside the cell, like the plasma and the interstitial fluid, about 1/3 of your fluid volume is there. Fluid balance. The body contains the required amount of water to meet bodily needs. Water moves by osmosis. The concentration of water in the fluids determines the fluid balance. Remember, osmosis is the movement of water through a semi-permeable membrane, like the cell membrane, from an area of greater concentration or percentage, however it's expressed, to an area of lower concentration or percentage. It's always moving from where it's more abundant to where it's less abundant. Now, water, the amount varies from person to person. Some factors are age, the gender, the amount of adipose. Fluid intake and output is determined by the diet, like the foods that you eat, dry foods versus like vegetables, which have a lot of water. Liquids that you may drink, like Cokes or teas or beers, whatever. Water. Metabolic water that's made as a byproduct of cellular respiration. We just don't make enough to sustain ourselves. And dehydration synthesis, when molecules are built. Remember, all the biomolecules being built by dehydration synthesis. Normally, the fluid intake is approximately equal to the output. I have in our notes around 2,300 mils per day. That'll vary from person to person. Regulation of intake and output. Fluids may be lost by the lungs and the skin. You can breathe on a mirror and you can fog a mirror up, or you can fog your glasses up when you're cleaning them. The skin from like sweat. Number 2 is vomiting and diarrhea. You can lose fluid volume on both of those ways. That fluid leaves your body. And fever and severe burns. Fever is going to cause you to sweat, so you'll be losing it by your skin again. And severe burns would form blisters. And that's going to be, those blisters are full of like water. Now you have a blank. The word that goes in the blank is electrolytes. electrolytes. Most solutes are electrolytes. They're compounds which dissociate into ions. Dissociate means they come apart when they're dissolved in water into ions, charged particles. Electrolytes dissociate in water to form cations, C-A-T-I-O-N-S. Those are your positively charged ions, like sodium is one in particular. Calcium is another one. And anions, those are going to be negative ions, like chlorine. Even the hydroxyl ion, OH, is a negative ion. Most are inorganic, and some are proteins. Now, non-electrolytes, compounds which share electrons, that's going to be the sharing of electrons, and don't form ions. Glucose and urea are examples of that. Some functions. Many are essential minerals. And I listed several here. Sodium, chlorine, calcium, potassium, magnesium, bicarbonate, phosphate, and sulfate. Those are all involved in making your biomolecules for sure. And for normal metabolism, normal pH and stuff like that. Control osmosis of water between body compartments. We'll look at that in just a minute. And help maintain acid-base balance. We'll see that also with our buffers. Now, concentration. Water moves to the area with the greater number of solute particles in the solution. More solute, less water. So water is going to move where it's more abundant to where it's less abundant. If there's a lot of solute there, a lot of dissolved particles in a solution, there's less water. Now I have a couple of examples here. Glucose is a non-electrolyte. Glucose forms a crystalline structure, which we call like sugar. And when you dissolve sugar in water, the glucose molecules just come apart from the crystalline form, and it's still glucose molecules floating around. So they do not come apart in water. They are non-electrolytes. They do not form ions. But you have sodium chloride there, NaCl, and you dissolve that in water to make salt water. And yes, the ions, the elements do come apart to form ions. There's your sodium cation and your chlorine anion. So two ions were contributed to the solution by sodium chloride. But look at calcium chloride, CaCl3. That's got 3 atoms there, and when that's dissolved in water, it dissociates or comes apart into calcium ions and chloride ions. So it has 3 times the effect on the solute. 3 ions. So we have calcium will also attract twice as many chlorines to its area than will sodium. Look at calcium, it has 2 pluses. That means it can accept 2 negatives, those 2 chlorines. Look at sodium, it's only got 1 plus, so it can accept 1 negative charge. Remember, opposites attract. Sodium has 1 plus, it can attract 1 minus. Calcium has 2 pluses, it can attract 2 negatives, 2 minuses. Now, distribution. There are 3 compartments you're going to find electrolytes in. Plasma, that's number 1, Plasma, contains many proteins, which are anions. Capillaries are not permeable to proteins, not large ones. So proteins don't typically leave capillary beds to go into tissues. Number 2 is interstitial fluid. That's your tissue fluid. Interstitial, interstitial fluid, contains very few proteins. Only very small ones can get out of a capillary. And then number 3 is intracellular fluid, intracellular fluid. The most abundant cation is potassium, and the most abundant anion is phosphate. This is going to be intercellular fluid like cytoplasm. We have this slide here, reabsorption of electrolytes and osmosis. So on your notes, we talk about sodium first. This is filtrate that's going through your kidneys. So it's going through tubules in your kidneys, and the little boxes on either side, those are cells around the tubule. So I have 3 tubule pictures here. Sodium is the most abundant extracellular electrolyte outside of the cells. It's approximately 90% of the extracellular cations. So the role necessary for the transmission of impulses in nerve and muscle tissue. Well, we did that in previous discussions, previous exercises or topics that we covered. Plays a large role in fluid and electrolyte balance. It says it's controlled by aldosterone, aldosterone, from the adrenal cortex. It causes the distal convoluted tubules to reabsorb sodium. So look at this picture here, and let me get my little hand out. And this tubule over here, this is the tubule that the filtrate that you're being filtered from your plasma is going through. And you're going to reabsorb some of that material that's in the filtrate. What you don't absorb is going to go out as urine. Well, aldosterone causes these distal convoluted tubules to absorb or become more permeable to sodium. So you see sodium is entering into these cells. So you're getting a high sodium inside the cells compared to the filtrate. Look at drawing 2 over here in the middle. Chlorine is a negative ion, and it's attracted electrostatically to sodium because it's a positive ion, And chlorine can go in and out of the cell membrane, so chlorine is going to follow, and you see that now you have a lot of electrolytes or solutes inside of the cells of the distal convoluted tubule. So you have more solutes inside the cells, fewer in the filtrate in the middle. So you have more water in the filtrate than you do in the cells. So the third image here is showing how water will passively follow from where it's more abundant in the filtrate to where it's less abundant in the cells. So the reabsorption of sodium by aldosterone causes chloride ions to be reabsorbed and water to be reabsorbed. Now, hyponatremia. Hypo means low, and natrium is sodium in Latin. So hyponatremia, Hyponatremia, excessive loss of sodium ions by way of perspiration, diuretics, or burns, causes muscular weakness, tachycardia, that's rapid heart rate, hypotension, low blood pressure, and headaches. Well, another electrolyte is the chloride ion. Chloride is mainly an extracellular anion. The role regulates osmotic pressure differences between compartments by being able to diffuse between intracellular and extracellular compartments. Remember, it got absorbed just a minute ago. It's also controlled by aldosterone, ALDOSTERONE, from the adrenal cortex. And chloride ions passively follow sodium ions. Now a condition, hypochloremia. That's written in your notes here. Abnormally low chloride ion levels in the blood due to vomiting, because what is the fluid of your stomach? It's very acidic, right? Hydrochloric acid. So you're losing hydrogen ions as well as chloride ions here. Dehydration and diuretics. Causes muscular spasms, alkalosis, and depressed respirations, breathing slower. Potassium, another electrolyte. Potassium is primarily an intracellular ion. It's the most abundant intracellular cation. The role says plays a role, impulse in the function of nerves and muscles. Remember the sodium-potassium pump? Sodium and potassium both are involved in nerve and muscle impulses. Plays a role in the buffer system inside the red blood cells, which we'll mention that a little bit, maybe in a previous chapter. Controlled mainly by aldosterone. There it is again, aldosterone. If potassium levels are high, then aldosterone is secreted and potassium is excreted. It's not reabsorbed by the distal convoluted tubules. So aldosterone is going to be involved in absorbing some electrolytes and actually causing others to be lost. Hypokalemia. It means low kalium. That's what potassium is in Latin. That's why its symbol is a K. Low potassium levels, possibly caused by vomiting and diarrhea, causes cramps, fatigue, and high urine output. Let's look at two more electrolytes here, calcium and phosphates. Now we know that calcium ions, or calcium, basically is stored in bones and teeth and can be released when needed by the body. Phosphate is also stored in the bones and teeth. It is necessary in the formation of DNA and RNA and for the synthesis of ATP and for buffering reactions. The role of calcium is an important structural component of bones and teeth. Well, we also know that it's involved in impulse conduction also of the membrane. It's also required for blood clotting, there it is, muscle contraction, and normal heartbeat. Controlled by parathyroid hormone and calcitonin, CALCITONIN. Okay. I always remember that it sounds like calcium, and then the last part is N. So calcitonin is responsible for depositing calcium into the bones and teeth. Parathyroid hormone, or parathormone, is secreted by the parathyroid glands. It's released when calcium ion levels are low. Okay. It stimulates osteoclasts to release calcium and phosphate ions into the blood. So the parathyroid hormone is going to be released released by the parathyroid glands when the calcium levels are low. Calcitonin, that goes in the blank, calcitonin, C-A-L-C-I-T-O-N-I-N, is secreted by the parafollicular cells of the thyroid gland. We saw the thyroid gland in the endocrine system where there was the parafollicular cells, which used to be called C cells for calcitonin. It's released when calcium levels are high. So you have high calcium levels in your bloodstream, calcitonin is going to deposit calcium back into your bones and teeth. So it's going to stimulate osteoblasts, the bone builders, to deposit calcium and phosphate ions into bones and teeth. Okay, one more, magnesium. Magnesium is an intracellular cation, activates enzyme systems needed to break down ATP into ADP plus phosphate plus the energy. It's also involved in reactions in the liver and the bone. Controlled by aldosterone. I picked a bunch of them that are controlled by aldosterone. Causes the kidneys to reabsorb magnesium. Now I have some movement of body fluids here, just to kind of give you an idea of how fluids are caused to leave capillary beds. It says exchange at the capillary beds is primarily a result of osmotic and blood pressure. Osmotic pressure and blood pressure. So see on the arterial side here, you have a higher pressure in an arteriole than you do on a venule. That also happens to be the case in the capillary bed. The arterial side of the capillary bed has a higher pressure pretty much when it's just coming off of the arteriole than when the capillaries join up to the venule side. So you can see that it says arterial blood pressure is higher than osmotic pressure. The net pressure is out. So you see that on this side, water can leave from where it's more abundant to the tissues where it's less abundant. Salts also leave. Oxygen, amino acids are going to also leave. And on there's your glucose. And on the venous end, this is where your carbon dioxide is going to be taken on from your cellular metabolism. It's the waste gas is going to be taken on back into the capillaries. Other wastes like urea and water can be absorbed in here too. And it goes into the venous side. So this is showing you how movement of body fluids is caused by pressure for one thing and osmosis another. You have in your notes overhydration, an increase in intracellular water concentration. This condition disrupts nerve cell function, causes convulsions, coma, and even death. So too much water can cause your body to have problems. Now water is good for you, but an excess amount of water is actually toxic. Acid-base balance depends on three mechanisms: buffer systems, respirations, and kidney excretions. So let's talk about this next little blank. It's buffer systems. B-U-F-F-E-R, buffer systems. A biological system which prevents drastic changes in the pH of a body fluid by changing strong acids and bases into weak acids and bases. Strong acids tend to contribute a lot of hydrogen ions. Strong bases tend to contribute a lot of hydroxyl ions. So let's look at some of these buffer systems kind of briefly here. Here's the first one we're looking at: carbonic acid bicarbonate buffer system. Carbonic acid bicarbonate buffer system. A weak base, and you look over here, here's a strong acid, here's a weak base, and a weak acid, here's a weak acid on the other example, are used to buffer a strong acid and a strong base. So here's a strong acid at the top, hydrogen chloride, hydrochloric acid, is being buffered with a weak base to produce salt, which doesn't have anything to do with pH, and a weaker acid, carbonic acid. Well, I'll put these little examples here showing the breakdown, how these will dissociate into electrolytes. Hydrogen chloride goes into hydrogen ions and chloride ions. The sodium bicarbonate dissociates into sodium ions and bicarbonate ions. The calcium ions and the chloride ions come together to form the salt, which is on the right side of the equation. And the hydrogen ion and the bicarbonate ions come together to form the carbonic acid, which is on the right side of the equation. Now we're going to come back to this weak acid in just a minute. Let's look at the bottom equation. Here's a strong base, sodium hydroxide, and a weak acid. Look at that, carbonic acid. That was that one right there. So an end product is used over again. This sodium hydroxide dissociates into sodium ions and hydroxyl ions, and the carbonic acid dissociates into hydrogen ions and bicarbonate ions. Well, the hydrogens pair up with the hydroxyls to make metabolic water. That's going to be a pH of 7, it's neutral. And the sodium binds with the bicarbonate to make sodium bicarbonate, a weak base. So a strong base was converted to a weak base. And look at this end product, this weak base, it was used to buffer a strong acid. So the end products are used over again. That's what's so cool about this carbonic acid bicarbonate buffer system. Here's another one that's very similar, the phosphate buffer system, and it works basically the same way. A weak base and a weak acid are used to buffer a strong acid and a strong base. And so you see that the weak acid is used to buffer a strong base. The weak base is used to buffer a strong acid. So it's the same type of a situation that exists here that existed with the the previous one, the carbonic acid bicarbonate buffer system. So let's move on to the next one. Hemoglobin, oxyhemoglobin. Oxyhemoglobin, this is right down here, HBO, oxyhemoglobin, gives up its oxygen in an acid medium, okay, and buffers the acid. Well, it's going to give up its oxygen in an acid medium, pH, and also when it gets down to your cells, it's going to give up oxygen to the cells. And now it's just a free hemoglobin molecule. Well, down at your cells, they're giving up carbon dioxide as a waste gas from cellular respiration, the metabolic processes, and that's going to come into the cell and into the plasma, combine with water to make carbonic acid. The carbonic acid is going to dissociate, come apart into bicarbonate ions and hydrogen ions. Now, hydrogen ion donor is an acid. So what's going to happen to that hydrogen ion? It's going to bind with hemoglobin. So hemoglobin acts as a buffer. Hemoglobin is acting as a buffer here. And in your notes it says the bicarbonate ion, HCO3, may remain in the cell and combine with potassium, or it can move out of the cell and combine with sodium. Remember the chloride shift in one of the previous chapters? In this way, much of the carbon dioxide is carried back to the lungs in the form of potassium bicarbonate or sodium bicarbonate. So we learned that hemoglobin can act as a buffer also. Protein buffer system. The protein buffer system is the most abundant buffer system in bodies, cells, and plasma. Proteins are polymers of amino acids, right? And you know that long chain of amino acids has an amine at one end of it and a carboxyl group at the other end. So I'm just showing you one amino acid here in our example. An amino acid is an organic compound that contains at least one carboxyl group and an amine, NH2. The carboxyl group acts like an acid and can dissociate in this way. So let's look at our first one where we see that the carboxyl group donates a hydrogen to buffer a basic condition. So in an alkaline situation, the carboxyl group can actually donate one of its hydrogens to bind with the hydroxyls to form metabolic water. Okay, so it takes the hydroxyl out of solution and doesn't let it make the solution alkaline. Well, look at the bottom left. The amine, on the other end, can accept a hydrogen to act as a buffer in an acidic condition. So there's a lot of hydrogens around. Well, this NH2 could take another one, be NH3. So it can remove hydrogen ions, so the amine acts as a buffer. Well, the amine can also donate hydrogens. The amine can donate hydrogen ions to buffer an alkaline condition. So it could donate a hydrogen to tie up a hydroxyl, again, to make water. So proteins are the most abundant buffer system in the body, and you can see why by these examples. Respirations, effects on pH. Well, here's the basic equation that we've seen time and time again. Carbon dioxide from cellular respiration combines with water to make carbonic acid, which dissociates, comes apart into hydrogen ions and bicarbonate ions. Now, the hydrogen ion levels can cause a problem. So I showed you, I'll give you 3 examples down here. In hypoventilation, where you're breathing slow, carbon dioxide levels build up in your blood, or in your body even, and as they build up, more hydrogen ions are produced, and so your pH is going to drop. That type of a situation is called acidosis. If it's a pH of 7.35 to 6.8, that's called acidosis. I've told you before the normal pH range is 7.35 to 7.45. Less than 7.35 is toward the acid range. 7.35 to 6.8, that's acidic, and it's called acidosis. If you're breathing too fast, like blowing up a lot of balloons really quickly, your carbon dioxide levels are going to be low because you're breathing real fast and you're getting rid of too much carbon dioxide. When that happens, your pH rises. It goes above 7.45. So alkalosis is 7.45 to 8. You don't have enough carbon dioxide in your body. Remember, you have to have a given amount. The partial pressure of carbon dioxide is supposed to be 40 millimeters of mercury to maintain your normal pH. So breathing is very important to the maintenance of your pH. You don't want to get rid of too much of the waste gas, and you don't want to have too much of it on hand. There's a sweet spot there that maintains your normal pH of 7.35 to 7.45, where your enzymes and your proteins will work in.